Okay, we've read our last speaker for the day and the conference, Mary Lou Gorno-Timpini.
Hello, thank you everyone. Thank you for sticking around. And thank you for the organizer for inviting me. I first of all wanted to apologize if I haven't been very social in these last couple of days. I've been really sick, so sorry for missing the dinner last night. So what I wanted to talk about today, it's a little bit different. I think we're going to be ending in a little bit of a different way. I'm a neurologist. I guess I would say I'm an anatomist, so we're going to look at quite a few patients, and we're going to look at their brains and sections of their brains, and trying to figure out a little bit what we know right now about the anatomy and physiology of the anterior temporal lobe, which we know is an area very important to semantic processing. So key points that I'd like to make. Focal neurodegenerative diseases. These are the diseases that I study, primary progressive aphasia in particular. They're neurodegenerative disease that affect very specific networks in the brain. In the case of progressive aphasia, networks that are involved in language. And they have a different lesion distribution than stroke. They don't follow the vasculature, so they give us a combination of lesions and network involvement that is very different from what we've studied in behavioral neurology, cognitive neuroscience in the last 2 centuries. So I think they provide a very good new lesion model. So language symptoms in progressive aphasia come from selective degenerations of nodes and networks. So the diseases start in one epicenter and they spread through, now we know, transsynaptic spread, across connected networks. So following these patients longitudinally gives us a very good idea of the interactions between different regions. And by studying these patients with a cognitive neuroscience approach, we were actually able to diagnose them better and identify different clinical syndromes that then correspond to specific molecules. So there is another level of analysis that we're not really used to with stroke. So with stroke, you have a lesion, the patient has a behavior, it was very relevant last century before imaging to figure out where the lesion was. Now is a little bit less relevant. But in these patients, the phenotypic identification and the association with anatomy is very relevant to diagnosis of what molecule is causing the neurodegenerative disease, because there are different molecules that can cause this problem, and the endpoint therapy for the patients is going to depend on which molecule is involved. So if analyzing the language symptoms and figuring out where the pattern of atrophy, which is not always obvious, is actually going to help us treat the patients better. So it has a whole different significance than just localization. So in the last, there's been a huge shift in thinking in neurodegenerative disease, a lot of new evidence that really shows that they're network-based diseases. So to give you an example, in Alzheimer's disease, the cells that suffer are in the hippocampus. In Parkinson's disease, you have specific cells that are target of in the substantia nigra. In behavioral variant frontotemporal dementia, which is basically the behavioral side of the progressive aphasia, you have these von Economo neurons that are involved. And these cells are in specific areas of the brain. So if you think of a specific area of the brain being susceptible to these diseases because it has this type of cells, then that will be the epicenter of that disease. And from there, these toxic proteins that deposit in neurodegenerative diseases, for instance, tau or TDP, we'll talk about it, they spread transneuronally to connected regions, so to regions that are connected even in the healthy brain. This is a figure from my colleague Bill Seeley. They actually won the MacArthur Genius Award for figuring out that in the different, these are also structural covariants in healthy subjects. So it's not just functional covariant. Really, if you look at the volume in this area and see whatever else in the brain, there are other regions in which volumes change in the same way in different people. You find the same networks that are involved in these neurodegenerative diseases. So it's a pretty striking story. So where does the susceptibility and vulnerability come from? Wide area E is the epicenter of that disease. So we think that there is a genetic developmental background for some reason. For instance, in a variant of progressive aphasia, we have a high frequency of dyslexics, so maybe that particular network has developmental susceptibility to disease. Environment, lifetime brain usage, we need to figure out. And so then we have this region that, because of these different reasons, has an increased intrinsic vulnerability to that disease. So when the disease starts, the first area that is going to be hit is that epicenter. And then from there, it will go to connected networks. So maybe to node 4, and then to node 3, and so on and so forth. So the disease of interest today, the disorder of interest, really is a clinical syndrome that was really described by Picks in the early 1900s, but it was introduced in the modern literature by Marcel Mesulam in one of his incredible endeavors, different endeavors. So it's defined as basically a progressive isolated language deficit. Patients are young, in their 40s and 50s, and instead of having a memory disorder, as we are used to in Alzheimer's disease, they have a language disorder. And here it was defined very vaguely: anomia, word-finding problems, and the anatomy was defined as left perisylvian. The neuropathology associated with it, so these toxic proteins that cause the neurodegeneration, were thought to be non-Alzheimer's. And that is really not true. We're not going to talk about it that much today, but there is a particular variant of the disease that is actually associated with early onset Alzheimer's disease. So many of the images that you'll see today are not activations, used to activation in these days. They're voxel-based morphometry, so they're areas of focal atrophy. So if you define the patients this way, with this very vague, not vague, but general description, the pattern of atrophy that you see looks like this. So the classic left perisylvian regions, but also these anterior temporal lobes that we're not quite used to seeing, and that we haven't studied as much as the perisylvian region because they're not hit in an MCA stroke. They're not hit in a middle cerebral artery stroke. So you have to have a massive vascular event to have involvement of the anterior temporal lobe, and you most likely will have involvement of the rest of the temporal lobe, not always, but most of the time. But isolated of the anterior temporal lobe is quite unusual. And that's why that area is being kind of ignored by behavioral neurology, cognitive neuropsychology for a while. Now, what we did when we started at the Memory and Aging Center about 14 years ago now, is despite being a neurologist, I had spent some time in London. I had decided that I wanted to put together a battery of tests with which I wanted to systematically test these patients, not just look at the symptoms, not just say they're anomic, but try to figure out the mechanism behind their deficit. And so by doing this, we were actually able to identify 3 variants. And some of them had already been described. The pattern in green here is the pattern of semantic dementia that had been described in England a decade before. But the kind of division of the primary progressive aphasia in 3 syndromes was kind of new. And today we'll really concentrate only on the semantic variant, which is too bad because I would love to show you the other ones too. But the semantic variant or semantic dimension is obviously the most relevant to the topic today. So each of these variants is associated with a specific behavior, a specific neuroanatomy, and also a high probability of underlying neuropathology, so underlying toxic protein. So the blue is the one I was telling you it's more likely Alzheimer's disease. So these are the patients you would want to send to a trial for Alzheimer's disease. They're in their 50s. They're very young. These patients with the non-fluent variant often have tau as their toxic protein, while in the semantic dementia, as we'll see in detail, the protein involved is called TDP. So this was highly debated, but I think there is quite consistency across centers now that these are the main variants of the disease. There are definitely other different presentations, mixed presentations. And after a few years of debate, we came up with a consensus or agreeing not to agreeing kind of idea. So I think if you read the literature about this if you become curious about this disease. Be patient that there'll be different nomenclature, at least until 2011. So, anterior temporal lobes, semantic memory deficit. After Rodrigo's talk yesterday, I thought we would spend 1 second defining what we mean by concept and semantic memory. We had a very nice spritz together last night before my fever came back and had a good discussion about the dissociation between semantic memory and episodic memory. So I apologize, most of you probably know this, but this is the main big division of long-term memory. And what we mean by semantic memory or conceptual knowledge is knowledge about objects and words and people that are not linked to a specific point in time. So you don't really know when you learn that a chair is a chair or that JFK is a president. Is part of your encyclopedic knowledge, is not linked to an episode in your life, and it usually doesn't have an autobiographical link to it. Although in the case of JFK, many of our patients remember where they were when he was killed, so in that case, that would be an episodic memory. And it's not that you cannot have an episodic memory about items for which you have a concept, but in patients, they really dissociate. And I think that was a little bit of the debate of calling those cells the concept cells yesterday, but I think at the end is just an issue of nomenclature. So that is in contrast with episodic memory, which is very specific in time and point and very autobiographical. So graduation, marriage, September 11th, specific episodes that are linked to an emotional content. Okay, so today we're going to talk more about this. Semantic information about objects, people. It doesn't matter if you remember Obama when he was young or after 2 presidencies, you still know who he is and know his biographical detail. Okay, so how do these patients look like? And I'm going to show you a few videos, so you don't need to follow all of this. But they have a selective deficit in semantic memory. Strikingly selective. They're in their 60s, they come to you, they say they have bad memory. But if you actually talk to them, it's not an episodic memory problem. They have difficulty coming up with words, great difficulty, but they're very fluent. They're different from the usual aphasic patient. If you ask them to name, they have a terrible confrontation naming problem. They have a single word comprehension problem. They have trouble identifying objects, people, sounds, really concepts. And they have trouble reading and writing exception words. It's very sensitive to frequency. This is a hard thing to get my colleagues, neurologists, to get away from just showing the watch and the glasses. They can do watches and glasses. You have to give them concept out of a little bit that are out of context. Huge effect of familiarity and prototypicality. Later, they have behavioral changes, and we'll see that this is very relevant to the anatomy of the temporal lobe. Striking sparing of phonology and grammar. So this is an example of the pattern of atrophy, selective atrophy of the anterior temporal lobe. This is all in neurological convention, so this is the left, this is the right. We'll talk about lateralization, most of the time bilateral but asymmetric. So let's see how an example of this patient, the effect of semantic loss on fluency. Pay some attention to the fact that he doesn't use many nouns. So he's very fluent, but he uses a lot of this and that and filler words and verbs, but not many nouns. So can I ask you please to tell me what you see in this picture and try to speak in sentences, please.
Yeah, well, this is a nice thing where I would be, right there on the water. That's a nice one. And of course, obviously, there are people who are just really seem to be enjoying it there. And they live there, at least temporarily. That probably is, they don't probably live there, but they just like to go there. And this guy and girl, they're having a lot of fun together. And
I think that gives you an idea. Guy and girl, here and there, they're having a good time. He's describing the picture, but without many specific nouns. This is actually his pattern of atrophy. Now, what happens to actions and objects? I think this is a good example. These patients do not have, they're not apraxic in the sense that they have the pattern of movement, but they don't associate it with the objects, and you'll see what happens here. But sometimes, since the semantic deficit is not an all or none, it's kind of a network degradation, giving them the movement of the verb actually can trigger the recognition of the item, and this is what happens here. Do you know what this is?
This is a demonstration of Flip for Mac.
What was that? How would you use it? How you would use it?
I don't use it.
Okay.
That's it.
That's fine.
Yeah.
That's fine, thank you.
Pink pearl. Huh? What do they use this thing for? Because I don't never
erase?
use this one. To erase things?
See, it's right
Oh, I
here.
see. Okay. Oh, yeah.
Yep. So in this case, me showing him the action actually helped him retrieve the information about the item. And this we see over and over again. So you show them a hanger and you say, what is it? I don't know. And then they say to hang. So it's quite an interesting phenomenon. Incredible naming deficit in which they usually give you the supraordinate of a prototypic or the prototypical item. Oops.
Is that a vegetable, a fruit? Fruit. Dog? I don't know what that is.
Okay, and this is a very typical clinical phenomenon. They're speaking fluently, you're talking to them, and then you give them kind of a low frequency word, and all of a sudden they say, I should know that. What is that? What is that word?
Over, and if you need flowers for somebody or something for presents, come on over and I'll give. Okay, now what's this?
Compulsion for
Take a
flowers.
look at that picture and tell me what you see, and try and talk in sentences.
Try to talk what?
In sentences.
What are sentences? See how?
So that typical, what are sentences? Right, one last one.
The cup. The cup?
That's right. How
Okay.
about the matches?
Matches.
Well done.
This one?
Which one is the matches?
I don't know what the matches are. My brain is asleep. These are flowers. I know that.
Show me the pencil.
There you see a little bit of
The
the
pencil?
behavioral issue that we'll talk
Yeah.
about.
Is that this one?
Good. Show me the flower
Right here. I knew the flower.
and the comb.
The comb. This one?
Good.
Okay, I think that gives you an idea of how they look like and how generally preserved they are in other aspects of language. So this gives you an idea of how the semantic representations kind of slowly lose their details, their specific features. So all the animals kind of become the same over time. Some patients can copy it very well or relatively well, but then they lose the specific features of the animal. So all animals become dog. They all become four-legged, kind of prototypical animals. They lose the ability to, same thing, the pumpkin becomes green because that's the most common color that you could use. I went to visit the Salpêtrière and I was struck to see how similar in French a patient is. I can't help but showing it to you.
Ça, c'est un chien quand même, non? C'est un chien, ça? Pas tout à fait, mais ça ressemble quand même à un chien. Ça ressemble à un chien. C'est encore un animal. D'accord. Pas sûr. Et puis après, il va aller où? Il peut rentrer dans une voiture, non? Se promener s'il veut. Avec...
So they have also this, it's hard to define, it is childish, a little bit detached behavior. So they know that they have problems, but they have a weird affect to it, and we'll get to that. The other amazing feature they have a surface dyslexia and dysgraphia, so they have trouble reading exception words. If we think about it, how to spell exception words in that nightmare of orthography that English has compared to Italian, you really need to memorize those words. They're part of your semantic knowledge, so they lose the ability of reading irregular words compared to regular words.
nerve, Sue, sword, shoe.
Bury, yacht, planel, tail, wolf, Iceland, wedding, chicken, colonel. What is colonel?
Okay, while they read
Snipe,
words normally.
Hans, Hoach, Smode.
She's bilingual German, so she has a little funny way of reading too. So, based on these patients, I think one of the most used scheme of how to think about a cognitive neuroanatomical model for semantic memory had to kind of include a hub. These patients have this semantic deficit that is being described as amodal and acategorical, and then we'll talk about that. But there definitely seems to be something going on in this anterior temporal lobe that does something, binds, converges, information that comes from upstream, modality-specific or category-specific information. And all this information gets somehow integrated in this area of the brain, and then we have our unitary concepts about objects and words, which is a little bit puzzling, thinking about the structure of this area of the brain, which we'll talk about. So this is another way of saying it. Primary sensory analysis, association cortices that do analyze spoken words, actions, written words, faces, buildings, and then all this information downstream, I guess, goes through the hippocampus. It gets linked to episodes, but then when the informations get consolidated, becomes a semantic memory in the anterior temporal lobes. Bilaterally is the system amodal, doesn't encompass all categories. These patients have been described initially as having an amodal and category-specific semantic deficit. But I think the story that is emerging is a little bit different. So first of all, we started to see patients with the right temporal variant of this disease. So the right temporal variant of this disease is a little bit, it's quite different. It doesn't start with that object knowledge loss, of word knowledge loss. It starts a little bit more as a behavioral syndrome or semantic deficits for people. So initially they were called prosopagnosic, these patients who have right more than left atrophy, but they're not really prosopagnosic because the deficit is multimodal. It's not just faces. They can perceive faces just fine. They just don't know who people are. And they have trouble from names and voices too. And in some way, they're less aphasic than the left temporal lobes. For instance, they don't have surfer dyslexia or have much milder surfer dyslexia. And then they have this striking behavioral syndrome, which they have loss of empathy for and coldness, deficit in emotional face recognition, hyperorality, and compulsions. So they develop compulsions for words, actually. They spend their days doing word puzzles because the word form and the phonology is kind of spared, and so they get fixated on language. The problem is that, as you can see in this image, for a while they came to us quite late because they were misdiagnosed as psychiatric cases. Years and years of couple counseling. My wife doesn't care about her children anymore and doesn't respond to emotional stimulus anymore. It's become cold, aloof. What is going on? And so they come to us much later. It's not a classic neurological syndrome as an aphasia. So this is an example of one case. It's not the classic double dissociation that you might see in other occasions. So it's a little bit more subtle.
Does this and this feel the same? I'm going to whisper something in your ear. Tell me what I say. Did you hear?
42.
Okay.
Yeah.
35. Okay. Now you can put your glasses back on. Do you want a Kleenex?
Emotional
Well, see,
dysregulation.
I get a lot of this water.
He's very sad, but in some way doesn't act
Yeah.
sad. There is this kind of
I thought I thought Kleenex.
dissociation,
Oh,
and
okay.
you have this uncomfortable feeling
That's why I get too much water in
when
my
you're
nose,
there
too.
because
Yeah.
there's really a mismatch.
I'll put these over here.
What is this for?
But then he still
Well, you
doesn't
just
know
hold
what
on
the
to it.
clinics
If you need them,
is
you
for.
use it. You can leave it there. I'm going to ask you to.
Okay. So when in a group of patients with these kind of problems, we correlate their ability to recognize emotional faces with voxel-wise atrophy. We get a right temporal lobe for recognizing negative emotions. We did an experiment in looking at different tasks on famous faces, so naming, semantic association. Keep in mind that in the semantic association task, we really match the faces so that really the association had to be made on biographical information that is mainly verbal. And then these recognition tasks. So the striking thing that they have about famous faces, for instance, the man you saw before, is that he has striking deficit on this test, but he doesn't tell you. It's not like a prosopagnosia patient that says, I can't recognize people. They don't tell you. And so it hasn't been asked for a while, I mean, for a long time in the literature. But actually, if you do the testing, they have a deficit. And I think the reason why they don't complain of it is relevant to maybe Rodrigo's talk yesterday, is that their episodic memory is spared, and they have this kind of, especially when it's a left temporal case, this visual pattern recognition. So when you ask them to recognize the famous person here, you don't ask them to name them or who they are because they have no idea. They tell you, you don't know, I don't know, but then they always point to the right one. So they have this type of kind of visual familiarity to the person. And when you do, again, a voxel-based morphometry correlation with this is in 170 patients with that task, you see that the semantic association and the naming are both left hemisphere, while this familiarity recognition is more right temporal. So that's why the right temporal cases have complained probably of deficit recognizing people, while the left temporal cases who have this spared can go about their lives using their familiarity recognition and episodic memory to recognize people that are relevant to them. So this kind of pattern has starting to emerge, in which we have a left temporal variant of the disease that is very that has deficit for words and objects more than social concepts, which they have surface dyslexia and dysgraphia. They develop an interest for a visual world. That's why I put those flowers next to that lady. She had her garage full of flower arrangements. So they get fixated, compulsive on visual information. And they have that kind of childish behavior that you saw in her. She was giggling. And instead, the right temporal cases have deficits for socially relevant concepts, faces, emotions, empathy, and they present more as with behavioral changes or with deficits for people, and they get interested in word games. So you create these asymmetries between the two temporal lobes. So what is the pathological correlate? Unfortunately, Mr. J, the man that you saw describing the picture, developed a compulsion for drinking alcohol, and he died of acute pancreatitis. The brain bank is right on the upper floor of our building, so I can walk upstairs and see the brain of the patients I've followed for 10 years, which is quite an experience, I can tell you. And here is his brain. I mean, if you look at it like that, you would say it's completely normal. And then if you start looking down here, you see this area that is a little, that is shrinking. Not the medial part. Medial part looks just fine, the fusiform and ventral. But this really tip and this area here that is very important that we will talk about. At the molecular level, the deposition protein is called TDP43. You don't want to know what it stands for, but it's a cellular toxic protein. We don't know what it does. There are genetic forms of it. We're making mouse model of it, and the whole thing. We still don't know how to cure it, but it's very specific and very different from Alzheimer's disease. Happens in the, it makes neurons die. This was actually discovered 5 years ago. Before, we used to call this the LDH, dementia lacking distinct histopathology. So it's a very novel and discovery, very fast-pacing field, fast-changing field. 98% of the patients in our UCSF cohort have this protein. So this is what I was telling you. If you can identify that an anomia in a patient is caused by a semantic deficit problem early on, you can make a prediction that this is the protein. And hopefully soon you will be able to give them a medicine for this protein and not for Alzheimer's disease. So it's a very relevant clinical issue, which looks really good on grants, by the way. So, anterior temporal lobe. What does it do? It always puzzled me. You know, when you study anatomy, the anterior temporal lobe is an allocortex. It's an old cortex. How in the world can it be doing this level of high conceptual knowledge that we're talking about? So I've always been curious about this. So let's go and look at it more in details. What is called here in green is allocortex, old cortex without all the 6 layers. This was defined as a multimodal region. And this superior and inferior really is modality-specific association cortices, visual and acoustic. This is a paper that I think is an amazing paper. I mean, I like anatomy papers, but it's really hard to read. It will take you, like, if you want to read it, you have to take aside a whole day. But I think it's a really fundamental paper to try to understand what the anterior temporal lobe does. And it really identifies that there are 7 regions in that area that go from limbic to paralimbic to neocortical. So I'll try to go through it quickly. The take-home point is, so this view, basically, it's a view, it's a rostral view, so you're looking from front to back. And this is lateral view. This is an inferior view, ventral. And this is medial. So here is the hippocampus. And basically, the 7 areas goes in the medial part. This is all paralimbic. The temporal tip, that is called TG here, is still paralimbic. So old cortex without very defined layers. The part that is really defined by von Economo as polysensory is this area here in the dorsal bank of the superior temporal sulcus and partly in the middle temporal gyrus too. So this is TG, that tip that is still allocortex. Acoustic, auditory cortex, high level TE is visual. And this part in the middle is the one that we call polysensory. The von Economo really called polysensory. So if you look at it in the coronal sections, they really look different. See how darker they look? And they look different because they have this very developed layer 2 and layer 4 in the cortex. And here we go, layer 4 and layer 2. This is again area TA, which is still for auditory, but polysensory. What does it do? It has these neurons in layer 2 where you get a lot of projection afferent and efferent to other regions of the cortex. So it kind of makes sense, right? If that's your semantic integration area, you want it to have a big layer 2 with a lot of neurons that communicate to other areas of the cortex. And then if you go and look in semantic dementia, well, look what is a disease of layer 2 of the cortex. So the TDP inclusions in this type C TDP that you don't want to know about, the important thing is that it's predominantly in layer 2. So it's really telling us that that's where, that's the dysfunction of the system that we're seeing in these patients in the early stages at least. So if we look a little bit more in detail of Jay's brain, I think it's really striking to see how really the visual part, there's been a lot of discussion that the ventral cortex is really the semantic region. And I think in some of the imaging studies, it's a little bit of an artifact, because if you have a lot of atrophy here, and I can assure you that some of this is gone. If you looked at it in an MRI section, it would just be empty. You can't correlate it. There is not enough variance there to correlate it with behavior. So what you're going to see is actually a correlation with the second region that is least involved. So in this case, his deficit, it was mainly in TG, so that little cap, but also going into the more lateral multimodal region. And I mean, we have to be careful. We know that the gross anatomy doesn't always correspond with the microscopic anatomy. So this is a study we did in collaboration with Brad Dickerson at MGH, I think the biggest cohort of ST patients ever seen, and he used the free surfer single subject approach a little bit like we used to do in stroke. So single subject, regions of most severe atrophy overlap, and the region that is most severely atrophy in 100% of the patient strikingly correspond with this polysensory, kind of auditory but polysensory regions. But this model that we've been using of this hub that connects to all the upstream regions needs this ATL to be highly connected. So let's look at that. This is again a study from Brad and MGH who used intrinsic connectivity arrest in healthy subjects to seed different regions of the temporal lobe. And you see that when he seeds in the anterior part of the auditory cortex, he does get auditory and somatosensory, but he gets more of a superior stream in the temporal lobe. When he seeds in the medial part, he gets the salience network, orbitofrontal, amygdala. And when he seeds in the more polysensory, he gets more of the default network, semantic network kind of picture. This is a study that we did at UCSF in which we seeded actually the site of most atrophy in the semantic dementia patients. And we were struck to see how this is resting state connectivity again. You do get connectivity with primary cortices, Heschel's, and then primary and secondary modality-specific regions. And actually in semantic dementia, when there is atrophy to this region, there is also dysfunction, changes in fractional amplitude, low frequency fluctuation, I need to read that, in the upstream modality-specific regions. So deficit in the lesion in the anterior temporal lobe causes a dysfunction in the whole network, not just in the anterior temporal, supporting that, that model of a hub plus distributed network for semantic memory. When we look at these regions that are connected and we look at the amplitude, actually their dysfunction in semantic dementia, these are sites far from the atrophy, correlates with specific cognitive tests. And in the right hemisphere is more emotional tasks, and in the left hemisphere is more word-based. Not only when we use atrophy in the superior kind of auditory regions or inferior visual regions, the level of atrophy in semantic dementia predicts the dysfunction on downstream visual and auditory cortices. Again, showing how much connectivity there is and how dynamic the system is, but not completely a modal. Then we did a structural connectivity analysis. This is a DTI study, and again, you can see that the medial parts connect more to orbitofrontal regions, so more of a behavioral social network, and the lateral parts connect to more visual and language regions. So this is a tractography depiction of that. So the arcuate fasciculus, very involved in language, is more lateral in the temporal lobe, and then medially there is inferior longitudinal fasciculus, which is a visual tract. And then more medially, again, there is the onsinet, which is a social behavioral test. The onsinet, the social behavioral connectivity, is larger in the right hemisphere than in the left hemisphere. And then to confuse things even more, semantic dementia patients are highly left-handed. So I think the overall picture is a little bit similar to what we heard for the visual Wolfram area and the fusiform face area. We think that the semantic system, we've thought of it as a modal supracategory system because most of these patients have bilateral atrophy, and maybe they have atrophy that goes to the superior and inferior part of the temporal pole, but there are patients that can have asymmetries, and there are patients who have different level of lateralization. So I think that the distribution of the neurodegeneration within the anterior temporal lobe and between the two hemispheres determines a variable phenotype. So it's basically a medial to lateral, right to left interaction, and it's not going to be 100%, but it's going to show asymmetric semantic memory deficits. And I hope I convinced you that these diseases are very good model to discover new brain behavioral correlations, and I hope some of you will want to collaborate. And I want to thank the lab, this is my lab, people in my lab at the Memory and Aging Center, and I'm sorry Kevin is with me now, Alfonso. This is Kevin Shapiro that worked with Alfonso for a long time. And Maria Luisa Mandelli does a lot of the imaging analysis, and this is the whole Memory and Aging Center that supports the whole research. It's been an amazing journey. We were 10 in 2001, and now this is the group. Thank you.
Scott, please.
Thanks for a wonderful talk. And along the way you had mentioned multimodal, and I was wondering if you could say a few things, because the examples were mainly in the visual modality with the patients.
Mm-hmm. Yes, no, the deficit is multimodal. It's just not 100% multimodal. So you see people, usually the most common form of the disease is the left temporal, so left more than right, and they have a greater problem with words, but they can't recognize sounds. And you saw when he was looking at that racer, he touches it, and he still can't recognize it. So it's very different from the classic agnosias that are tactile, auditory, or visual. This is from all the modalities. But then he still can access the semantic information from the action. So it's not that, it's not 100% multimodal, I think.
Thank you for that lovely talk. I have a question for you, just following up on the action knowledge of these individuals, which seems totally fascinating. So you mentioned that they preserve the knowledge of how to use the object, but I'm wondering, have you or anybody else characterized their knowledge of the meanings of the words, aside from, of the verbs, right?
Yes.
Aside from being able to do the motion, and is there a verb-noun dissociation, an object-action dissociation? What does that look like?
Yes, I think there is a verb-noun dissociation. It's very difficult to really prove it because they're so exquisitely sensitive to familiarity that it becomes very difficult. But it has been shown, for instance, they produce more verb than words. Usually the two things go together. If they don't know the object, they don't know how to use it. But then they can use affordances. I'm not sure, I ask you action experts. If you put the thing in their hand, usually after a while they can figure out, they can show you the right action because the knowledge of that action is there. It's just not linked to the object anymore. But experimentally, it's very difficult to show because it's so sensitive to frequency, and then it confounds everything.
So I guess what I'm trying to get at is it just the object, or is it that their knowledge of the motor program or the action sequence is dissociated also from the meaning of the action itself, right? So do you know what I mean? Because there's more to, like, the action to give or kick or exercise has more to its meaning than merely some motion. And so my question is do they have preserved, praxic knowledge, but lacking?
They don't have preserved abstract knowledge of the verb. It's just the motor program somehow. That it's kind of, I think, the same mechanism as that familiarity for the face, right? They have that, I don't know how to define it, almost procedural. They have that action plan there, even if they don't know what the verb means anymore or the object means anymore.
Thank you. That was a lot of great research. I have a question about the lateralization of objects and social knowledge to left and right, respectively, as you were saying, and also the dorsal versus, or I guess lateral versus medial dissociation between limbic and the other structures. So there have been a few reports now in the literature about reading social words, reading social emotional words still activating left superior temporal sulcus, all along the sulcus even, but extending fully into the anterior part of the temporal lobe. And so if these social words are still activating left side, and they're activating STS and not just the medial parts of the anterior temporal lobe, do you think that this challenges your assertion that there is this object to the left, social to the right?
Well, I don't think it's not as simple as that. You know, in that case, I would say they're words. So there is that kind of interaction between category or type of knowledge and the fact that it's verbal that still strikes it to the left. And that's why, you know, patients with left temporal atrophy have trouble with emotional words. It's not that they don't, but then they have a little bit less trouble with the emotional faces, because that you're kind of putting the two together, and the right hemisphere is wired more for emotional faces. And maybe that's what I would have said to the talk yesterday. I'm not sure that the lateralization is really driven by language, because social cognition and social value is even, you know, appears earlier than language. So maybe it is actually that drives the right-sided specialization. There is this intriguing finding of high frequency of left-handedness, and you know, these patients also have high frequency of inflammatory disease, and there is this old concept in the literature from Geschwin that inflammation in the womb can determine handedness, and we're kind of looking at the lateralization genes to see if part of the pathogenesis of this disease is actually a problem with lateralization.
Thanks.
Laurel, Alfonso first, and then it's going in order.
I have the way in which you present the data is showing. You said there were 70 patients in one case, 100 and something in the other case, and that's great in one sense, and it's terrible in another, because you're putting together all kinds of things that may not or should not go together. So if you were to take an individual patient and you were to sort of longitudinally look at the pattern of performance, what is the pattern? That is, you make it sound as if there is this network, there is this thing all hangs together, but perhaps that's misleading. Perhaps that's the consequence of lumping together 170 patients, rather than looking at the pattern of dysfunction that emerges as the disease develops. Do you have any results that can illuminate what is going on really?
Yeah, no, I mean, we do love to do single cases. And I think the whole concept of the right temporal variant came from single cases, and we're still working on single cases, and it's striking the kind of unusual semantic deficit they have. So one of the ones that I described started as a semantic deficit for food. So very kind of, right, and smells, so non-verbal type of knowledge. So I think the idea actually came from single cases, and now we go back to our, you know, they're not that frequent, right? Even in a center like the UCSF, which is a tertiary referral center, we get about 20 per year. So now that we have these hypotheses that came from the single cases, we can go back and look at the pattern of anatomy in all of them. So initially, they have this kind of modality, more modality-specific deficits, and then longitudinally, they become more of a multimodal.
Sorry, may I? But that's the point. The point is that if, in fact, you are looking at something that has evolved into this complex picture, that is giving rise to a very different interpretation of what these areas do. That is, in fact, there is structure in its system, and it may be so that the notion that it's multimodal or amodal may not be correct, for example. In fact, it may be a whole bunch of things that happen to co-occur, and this disease may not be the best way, other than longitudinally, for looking at the ways in which the system is structured. And that's because, in the end, what you're going to do is you're going to have a pattern of associations. But the associations are a mere consequence of the spread of the disease across different areas, as you pointed out. So it seems to me that it's fundamental in this case to do the studies longitudinally and see how they segregate into the parts that comprise the whole.
Definitely.
Well, believe it or not, I absolutely agree with Alfonso. So I was going to make actually a similar point about the disease progression and the importance of characterizing the stage at which you're capturing these patients. Because as we know, it does progress from anterior, tip of anterior pole, and the more involvement, the more late in the disease, you get more and more posterior spread of the disease. And in some of the patients that you showed us, they have a lot more than just an aphasia, right? So they have an agnosia. They no longer, by anybody's criteria, they do not know what those objects are anymore. Right. So, and this gets to Marina's point about, or question about, how are they with actions? How are they able to, you know, part of the problem is if they don't know what the object is, of course they can't evoke the action that's associated with it. And so it's a very difficult to test for the integrity of the action representation until you can somehow get them to activate the sufficient amount of object knowledge. The other point I wanted to make is about the notion of an amodal hub in ATL, or multimodal hub in ATL, and the fact that that may be one of the semantic hubs in the brain, and that there's another very important semantic hub in the posterior temporal lobe. And Alex Martin has actually written about this, the importance of this tool hub, and Marius Pielen's work speaks very strongly to this, that there's sort of another hub in the back of the temporal lobe that's really important for coalescing our tool and manipulable object knowledge. And so, consistent with that, in these semantic dementia patients, you often see a very interesting dissociation between their animal knowledge and their tool knowledge, and that tool knowledge gradually degrades as the disease progresses.
No, the tool knowledge is there from the beginning. It's lost from the beginning. So let me show you. I don't agree with that. I'm sorry. I know I'm
okay. I don't remember. There are some. I wish this is not really my area, so I don't remember the citations, unfortunately. But I know there's.
No. But just in the patients. I'm not saying. I'm just telling you what happens in the patients. So the patients who have selective atrophy out here, they don't have the action schemes anymore. Actually, I have a video to show you. But they know what the objects are. They know what tools are. And it's just that they don't know the action scheme that goes with them. So they tell you a light bulb, or you use it to turn the light on, it goes up there, and then they go like that.
Right. But if you test their...
But they know the object.
Yeah, but if you do sensitive measures like RTs to do animal versus tool naming, they're slower and have more difficulty with the tools. And they make more errors. They make more thematic errors. That posterior group makes more associative errors than taxonomic errors. So there really are differences in the semantics of those two groups and the way those two semantic systems are organized. But we can talk more about it later.
Sort of a follow-up question. If you look at the anteriors at their in-home, what are they like in terms of their action sets and behavior?
So on the tools that they use every day, they're fine until they lose the knowledge of what that thing is, and then they don't know what to do with it anymore.
Yes.
So there is this striking interaction. It's very hard to test things in them. Like I try to do semantic features, and then they don't know what the word of the feature is. So you ask them, well, does an elephant have a trunk? And they say, what's a trunk? So then you can, so I would love to have you guys help to think about how to do it.
Rita, let me jump in just because you've already asked one question, and let me go at this from a different perspective. I don't doubt any of the data. A lot of us look at the imaging data, and I'm talking about the PET data, for example, where you don't have those difficulties. And you can see that whatever people mean by the anterior temporal lobe, the hypometabolism clearly extends throughout a large part of temporal cortex. This is my confusion. I look at that I thought that Ding paper, that's the anatomy paper, was fantastic, because what it showed was that if you just look at the pole, there are, I think, 6 or 7 clearly defined areas based on the cytoarchitecture. As you move back, like every place else in the brain, there is all this structure. Fusiform, inferior temporal, middle temporal, amygdala, on and on and on. Why is it that in this part of the brain we abandon all that thinking because somebody came up with a notion of a semantic hub, which I still quite don't understand? And let me just give you an analogy. Let's take a disorder of posterior temporal cortex that blows all that tissue out, lots of pathology, and what we find is this kind of agnosia pattern, and I argue that this is a perceptual conceptual hub. I think most people would say, well, what are you going to do about the fact that we now know all that stuff we've been hearing about face areas and word form area and so on? Independent of whatever way you want to discuss posterior temporal cortex, we're going to discuss it within the context of the differences in anatomy and the differences in function. Why is this area of the brain, why don't we think about it the same way? Why do we dismiss all the anatomy to then turn to this other concept, which I find extremely hard to grasp? What's a hub? And why should it deal with everything?
Well, I don't know what's a hub. I thought you were going to ask me the nature of the representation, which is even harder.
But it is very confusing for a lot of us, certainly for me.
Yeah, no, it is. But I mean, there is this area right here that is polysensory. It responds to many modalities. So I think there is something going on there that integrates the different modalities, and that is the neocortical area that has that huge layer too, with huge intracortical connectivity. So I think there is something there that makes that patient not knowing what that eraser is, not knowing the name, not knowing how to touch it, not knowing how to use it, that is different from a visual agnosia that comes on from a more posterior region.
Oh, yeah, no. The issue is not that it's not, it should be different. And the issue is not whether it contributes to conceptual knowledge and semantics. I think what a lot of us struggle with is what's the contribution and why does it have to be termed as a single thing? Okay. One last point before we continue. The data that Laurel was talking about, there's now 4 studies in the literature with damage to posterior middle temporal where the claim is that, not the claim, the data show that there is loss of information about tools across modality, across function, whatever you want to ask them. And, like in Tim Chalice's study, they had quite a number of patients with anterior temporal tumors who were fine on their tests. So that's where this idea comes. Well, if you want to have one, maybe you're going to need a lot of hubs.
I agree with that, and that's why I showed you the functional connectivity study, which I had to go through quite fast, that actually shows that these regions contribute to semantic processing. They do. They just don't create that profound kind of deficits that you see in the anterior temporal lobe, but they definitely contribute. And I have, do you want to see a video on an LPA patient doing an action? This is impossible. Okay, let me see. Talks. This is not the usual resolution. It doesn't usually look like that. is.
Use a light bulb.
Put it up there, and then it's about like that. Holding it.
And what do you do with it?
And then I put it in the stuff, what do you call it? I forget what it calls. Going up into the thing there.
So this is
So
what.
pretend you're holding a key. Show me how you'd use that.
To put it into a thing.
So show me the movements. How would you do that?
Put it in there and
All right. Pretend you were holding a sponge. Show me how you would use it.
say it again.
A sponge.
A sponge, yeah, so it's
striking how young
using
these
it to
patients are, huh?
wash them, isn't it?
So I have no doubts that having this type of problem is going to slow her down on concept of objects. But it's just a different, in my way, it's a different type of it's more specific to action, and then action is so important to knowledge of objects that it's going to make knowledge of objects impaired. But
directly to this example, is she unable to reproduce the kinematics with her own hand, or would she also be unable to recognize the kinematics if they were represented to her?
This is the
So
study
if you
that
would-
Miguel is doing, and this patient in particular, I don't know, but I think she will be able to recognize it, and she will do it on imitation better. It's just the doing it herself that is more difficult. But I'm not quite sure. This is a brand new study that we're doing, so I'm not quite sure how she did in the recognition. We're doing a pantomime recognition and a whole battery of tests. I'm not quite sure how she would do it, recognize it. Maybe you do. In the pantomime? Mm-hmm. Yeah. And actually the non-fluent, so we did, I just forget it. I wanted to show you a whole other thing, but I can't. On this more action knowledge. Did I put a slide at the end maybe on it? No. Anyway. The frontal patients have trouble with action knowledge.
Just to clarify, the issue is not necessarily whether they have those difficulties. The issue was relating it specifically to the anterior temporal lobe as opposed to more widespread and involvement of lots of different systems. That's all. Like posterior temporal.
I totally agree with that.
That's, then you don't have
It's just striking that she knows what the light bulb is, right? So she doesn't know the action, but she knows what the light bulb is. While the anterior temporal patient wouldn't know what the light bulb is and also doesn't know the action. But then you give her the light bulb and she'll go like this. So they're just a different type.
I know that these semantic dementia patients, not all of their deficits seem to have to do with labeling and language, but it always strikes me looking at these studies how often the task, and understandably we use language to explain these experiments, how often it seems to be involving the label or accessing the label. So I wonder if you have the idea that it may be the extent to which the label allows them to access the rest of the representation. So it's more having to do with the language and the label rather than the rest of the representation being damaged. Is that consistent with what you're thinking about?
Well, they have difficulties with tasks that don't involve, like the typical task that we use clinically is called the pyramid and palm trees task, and it has association only on pictures. And even if they don't have to come up with the name, they have trouble. But the name is definitely the first thing they lose. I think it's just a more kind of labile semantic feature of an object, right? The action has all sorts of semantic context to it, while the word is a very fragile link feature, and so is the one that is lost first. They have unbelievable naming problem, like 2 out of 60. It's basically diagnostic. There's no other category of patient that does 2 out of 60 on the BNT, and it has that fluency and that grammar and that.
With the chair's permission, can I ask you a question? Which is, why did you end your remarks just then by kind of exasperatedly saying, well, then you don't have a hub? What did you said, well, then you don't have a hub. Why would the presence of a medial temporal lobe tool system mean that there isn't an anterior temporal hub? But I mean, you know what a hub means in United Airlines, and there's multiple of them.
Laurel's point, and the point we were trying to make, is that there seems to be multiple hubs. Well, the model here is that there is not multiple hubs, there's one.
Right, but it seemed like you were trying to, I interpreted your...
No. Well, I wanted to dismiss the idea of one hub.
Okay, there's not...
They're all hubs, yeah, there are lots of them.
Well, they're all nodes. I think there are nodes, and there is a bigger hub. There's an action node.
So this is a really interesting discussion. Just to take it kind of further a little bit, or rather on the other side, when you say that there are auditory and visual regions in the anterior temporal lobe, I'm trying to understand what you mean by that. Because when you showed us the connectivity data, the so-called auditory region was actually connected to like a third. I mean, it's to lots and lots of things. A subset of which was potentially auditory cortex. I don't know is there some electrophysiology data? What do you mean by that, and how does that relate to what people mean by the auditory cortex being the auditory cortex?
There is a lot of data in the monkey literature. It's hard to do in humans, but there's a lot of data in the primate, non-human primate literature saying that the tip of the superior temporal gyrus, it's an auditory area, high-level auditory area, and all the tracing done by Marcel Mazelon and his group showing connectivity to Heschel gyrus. And this is the intrinsic connectivity on fMRI is a very crude method in some ways. So you don't know if this is then connected to this because of the time resolution. So I want to show that it is connected to auditory cortex, and then from auditory cortex you have a resolution, the lower resolution of fMRI, so you can see activation related to that too. That's why I think all these connectome analysis need to have really specific hypotheses, otherwise you find whatever you find.
But I guess my question is there any evidence in humans that there's any auditory or visual or anything subdivision in this part of the brain? That might be hard evidence to get, but I don't know what
it's hard, and it's a hard area to get to. This is why these patients have been so important to study. fMRI has a lot of artifacts there. We all try to do our best with that area, but it still has a lot of artifact. MEG doesn't get to it. Stroke doesn't get to it. It's interesting that the epilepsy patients, the temporal lobectomy patients, don't have the same semantic deficits that these patients have, but I think it's because they're more medial. What they take out is the more medial part. That lateral kind of polymodal area, they don't take out. So I would take these as just patterns. This medial part is much more involved. It looks different from this, but from then on, it's difficult. It's a very new area to start, and I agree with Alex that we need to, we don't need to think it as a whole unitary region. We need to go beyond that a little bit of just a big blob that does everything, amodal, acategorical, and everything. I think that's not the end of the story.
So, just if we could stay just with this slide for a second. The thing that doesn't make sense to me about this picture, with the discussion with respect, for example, to this kind of lateral MTG yellow highlighted spot, being polysensory, right?
Yeah.
Let's say this is the conjunction point, right, of the different sensory systems that we would typically think about, the visual system and the auditory inputs, and everything is going into this one spot. That picture that you're showing does not fit with this at all, right?
Yeah.
I mean, you would expect, you see this, you get both vision, you get all the senses
Well,
back,
that's where you
right?
get here is a different method. This method is not sensitive enough. This is just the seed method, while the FALF method here, alpha method, is more sensitive, and then you do see it.
Well, the alpha is not connectivity, though. That's just the local amplitude of fluctuation.
Yeah,
power.
but you look at how they change together. So here you do get connectivity to sensory, motor, and visual. I think that was the first time really that we were able to show
But
it.
just to follow up a little bit. So what you would like to be able to see, if this really is a hub that is supposed to work in the way that it's supposed to work, we need some better methods or something to show pretty transparently that this is the picture that actually comes out, right? And unfortunately, we don't have tracer anatomy.
Right.
We don't know how to relate this to the monkey anatomy that we
Right.
do have tracer anatomy. So those are the kind of problems. Now, it'd be great, now DTI to the tractography to the extent that this is possible, and maybe it's really not in using it this way. I know there's a lot of criticism of that method,
Right.
trying to do track tracing. But ultimately, what would be great is if you could show, or somebody who has these kind of data could show, is you take these different cytoarchitectonic divisions and actually show that the DTI data would actually flesh that story out, such that that more superior aspect would actually go with the right cortex, and that this
Right.
probably would
Which
actually
is
show
kind of
that. Because that's what we don't really have, to be able to put that whole picture.
This is what we tried, but the problem is that the resolution is not as. Now I'm trying to get my neurosurgeon colleague to do some stimulation of the different areas of the temporal lobes doing surgery. I think that would help us. This is our kind of rough trying with DTI. I think this experiment. Sorry, I'm going backwards. This experiment with Bill is the one that got us closer to it. Showing that there is this connectivity with primary and secondary association, primary and secondary unimodal regions, and that the FALF in that region actually correlated with the semantic deficit. So to show that all the hubs are working together.
Is there time for one more? So this is not necessarily a cognitive neuroscience question, but I'm kind of fascinated by it, at least for the kind of pictures you're showing and the idea that there might be some kind of almost like viral spreading of the pathology, by axon, something like that.
Yep.
What's always kind of puzzling to me when I look at
Prion-like.
the atrophy patterns, and you got these different kinds, why on earth would it be lateralized? There's clearly system specificity to the different disorders, right?
Right.
And the different patterns.
Right.
So there must be some differential kind of biological markers that help mediate that. But is there actually something like a protein distribution or something that's actually, why do the left people get the left and why do the right people get the right?
Right. Yeah, I wish I could answer that. We get into that. I think we're doing a genetic study on the language genes to see that why in the world these patients with Alzheimer's disease present with a progressive aphasia when they're 50. Now, as I said, one thing that we found is that 50% of them had a history of developmental dyslexia. So I'm not saying that dyslexia is a cause at any rate or even a risk factor, but if you have dyslexia and you happen to get this disease, it might show up as a language problem earlier, right? Because that system is already, it was like that first slide, the susceptibility. So we're finding more and more about that. There is a version of this disease, it's called posterior cortical atrophy, that is very visual spatial. And they seem to have a history of dyscalculia. So these things we never ask in adults, which we really should. So I think it's going to be a combination of genetic, environment, but it's a very fascinating way to look at susceptibility. And now that we've discovered a few genes, we're going to study carriers very early on. Unfortunately, the genetic form of the disease is more severe, is more widespread, so you won't see that kind of focal atrophy.
We just have a couple of housekeeping issues to deal with, but before we do that, let's thank Mary Lowe for fantastic. I think Jerry has a quick something to say.
So last night Sharon said that this was the most important meeting in the year in her calendar, and in fact in many other people's calendars. And I wanted to point out a couple of reasons why it is. One of them is the vision of the organizing committee, and Alfonso in particular, to invite so many people from different traditions and different accounts and different theories. As far as I can tell, they're all not Alfonso's theory. But related to that is the other thing that makes this such an important annual date, and something I wish we could see elsewhere manifested as well as it's manifested here, which is that, again, through this vision, the committee, and again, Alfonso, Alex, and Mel and others, have managed to put the fun back into scientific agreement and, importantly, disagreement. And so for that, I want all of us to thank the organizers for a fantastic meeting.
All right, while we're all applauding, the meeting doesn't happen by itself. Everybody knows that Marius does all the heavy work, but he's not here to receive his applause. We only have one student here. He's going to have to be the representative for us to thank these guys who run around the auditorium when we point as if they're like remote control robots. So thanks to these people. And maybe one more thanks to all the speakers who did such a fantastic job here. We appreciate tremendously that they came. So. And finally, you all know that next year's our 10th. You got to start thinking fast and hard and send all of your recommendations to Marius because, like I said, he does all the work for us. And finally, give yourselves a great round of applause and see you next year.